First of all, thank you very much for the invitation to speak. As it turns out, I am speaking from Canada. I'm in Halifax, Nova Scotia. I had intended to go to Montreal and then to go on to Halifax to stay with my daughter and family. So I am now quarantined in Halifax and delighted to have the opportunity to speak with you. I'm going to focus on water-related disease, cholera, initially, in order to set the stage for the work that I'll present on COVID-19 at the end of my talk. Obviously, waterborne diseases are really a major problem in the sense that millions of people die each year, particularly from diarrheal diseases, which includes cholera. Cholera is a global disease. It's a water-related diarrheal disease. It occurs in pandemics. The first started in the 1960s, and it now occurs in more than 50 countries, affecting about 7 million people. We've unfortunately associated cholera with the Bengal Delta, Bangladesh and India, but in the United States and in Europe, we had cholera until the 1920s when we began chlorinating water. What I want to point out is that the cholera bacterium is a naturally occurring bacterium, and it's highly unlikely that we can eradicate cholera, but we certainly can control it. We can culture the bacterium, shown in the slide in the upper right, easily on media, as shown in the upper left. And the cholera patients are monitored by the fluid loss. And the work that I described began in the Chesapeake Bay and in earlier work in Washington State in the US. These are the samples that stations that we studied. Now, the major finding in those early years was that the cholera bacterium is associated with copepods. It's associated with this zooplankton and in the world oceans. This is the culprit, so to speak, the copepod host. The early work we did back in the 70s showed that there's a relationship between seasonal events with warming in the springtime. This results in algal blooms, phytoplankton blooms, followed by the zooplankton blooms, the copepods feeding on the phytoplankton, and then the crash of the zooplankton blooms with the release of the Vibrios into the water column, hence the transmission for those who drink untreated water. Now, in the Bay of Bengal, where we did most of our work in the 1970s, 1980s, we worked in the remote villages. Here you can see villagers collecting water, but these ponds serve as drinking water. They serve as the ablutions and for washing food and so forth, but also, unfortunately, in the far corners as the latrines. Sure enough, the model that we had developed early in the United States in the Chesapeake Bay and off the coast of Washington State held here as well, in that obviously there's person-to-person transmission, which is a very important factor, but ultimately the source of the bacterium is the environment, and I think this needs to be understood. Now, it occurred to us that phytoplankton could be measured by Landsat satellites. The Landsat was launched in the late 70s, early 80s, and so at that period of time, we did a very crude analysis using satellite sensors, and we were able to demonstrate a direct relationship between sea surface temperature, the lines in blue, and the cholera outbreaks in Matlab, Bangladesh, just off the coast of the Bay of Bengal. Now, we perfected our model and showed the very significant and positive relationship between cholera, chlorophyll, and rainfall. It was sufficiently accurate that a milligram per cubic meter increase in chlorophyll measured by satellite would result or be correlated with a 33% increase in the number of cholera cases in Bangladesh and in India. And similarly, a millimeter increase in rainfall led to about a 7% increase in the number of cholera cases. This held for both Calcutta, India, as well as Matlab, Bangladesh. So that was the theoretical framework for predicting cholera outbreaks in epidemic regions. Early, 4 weeks early, very high temperatures, warming, blooming of plankton, then a heavy rainfall, and where there's not good water safety or good security, there's a high cholera risk. Now that was the work back in 1996 using Landsat. Since then, we have gone on to use a series of satellites and to compile the data to improve the model. And in fact, we reached a point where, as shown in this case with Hurricane Matthew, which traversed the coast of Cuba and Haiti and up into along the coast of the United States. From the data, we were able to develop a prediction. And indeed, we predicted following the Hurricane Matthew, you will see on the left-hand side, the areas where we would predict cholera, and on the right-hand side were the actual cases. So this then was a demonstration of the effectiveness of our model. Now, let me just go back. The prediction for Matthew, we carried to Yemen. And we were able to predict in 1917, using the same model, cholera in Yemen. We wrote up a paper. This was published and a squib was prepared for the Scientific American. And a colleague in England read the Scientific American, and in January of 2019 called us and said, 2018, and said, I'm sorry, in 2018, and asked if we could collaborate. Indeed, we did so, and so we now have worked out a program where we provide a 4-week ahead of time prediction of cholera in Yemen, and the British Aid Agency and the UNICEF now locate physicians and supplies where we have predicted the highest risk of cholera in that part of Yemen. And we were able in 2018 to reduce cholera significantly. Now, another point I'd like to make is that the relationship of the environment and cholera can be demonstrated to be affected or influenced by climate warming, global warming. This is a series of analyses of plankton collected between 1958 to 2011 and to the present. And the red squares indicate the samples where plankton was collected. Now, this was a unique collection. This was what you would call citizen science. These plankton trapping devices were sent to ships captains, yachtsmen, to ferry boat captains, cruising ships, and they were asked simply to collect the plankton and send to the British Marine Station in Southampton, England. And when we plotted the data, it was fascinating that we were able to show that there was a relationship with warming temperature in the sea surface and the increase in the numbers of Vibrios. These Vibrios are the ones that cause disease: cholera, Vibrio perihemolyticus, which causes food poisoning from contaminated fish, Vibrio vulnificus, which causes a severe infection, which can be lethal, a 58% mortality rate. So we were able to correlate with the increasing number of cases of infection in Europe and England with the increased numbers of Vibrios picked up from the plankton samples that we had gone back and with molecular genetics had been able to quantify the Vibrios in those plankton samples from 40 years ago to the present. So a demonstration, I think, the first of using these kinds of data of a global warming influence on a human infectious disease. So I think this, is an important step forward. Now let me turn to work on genomics. We've developed a technique over the last decade or so of being able to extract DNA from whole samples. The DNA is then sequenced, and the raw sequence reads, we match up with a database that we have built of about 160,000 microorganisms, bacteria, viruses, fungus, and protists. This allows us then to determine the presence and identification of everything in the given sample that we started with. Identification of the bacteria, the viruses, the protists, the fungus, and as well to determine the genes they carry. This allows us then a very precise identification and characterization. This allows us to identify all the way down to strain and substrain. Very important because, for example, there is a species of a bacterium, Lactobacillus casei, but one strain is used to make cheese, another strain is used by industry to make Chardonnay, and a third strain is used by the various yogurt manufacturers to manufacture yogurt. For physicians, strains are very, very important because those bacteria in our gut of E. coli, we all have E. coli in our gut, are essentially the good strains in that they produce vitamins and they protect against other pathogens. But the one strain, 015787, of course, causes hemorrhagic enteritis, and that can be fatal. Here's an example of why it's important to understand strain. In this case, a young woman built herself a zip line. She was using the zip line. She was a 16-year-old. It broke. She fell into the water, gashed her leg. It developed into a necrotizing cellular fasciitis, from which was isolated 4 strains of bacteria. These were sequenced. 3 were found to be clonal, that is, practically identical, and the 4th was different. When we did our experiments to reproduce the infection by injecting mice with all 4 strains, we found that only 1 strain went to the liver and spleen. Now, this was very astounding. So we repeated the experiment by first engineering that 1 strain to be luminescent and to be kanamycin resistant. We re-injected the mice, and then we found that indeed it was only kanamycin resistant luminescent strains that went to the liver and pancreas. So here is a demonstration where it was very important to understand the strain identification. As it turns out, the 1 strain produces a toxin that allows the strain to go through the tissue to the liver and to the spleen. Now, let me turn quickly to water safety, and then I'll bring all this together in a moment. We've been working with Orange County, California, where, because as you know, the previous droughts led to water shortages. And so the sewage water was piped to the drinking water plant and then very intensively treated in order to provide safe drinking water. We did analyses with the Orange County water treatment plant, taking samples after microfiltration, reverse osmosis, etc. And we were able to show that indeed, when the sample comes in from the sewage treatment plant, that gray area encompasses all of the many, many kinds of microorganisms, including some pathogens and many not pathogens, but a lot of microorganisms. But after it goes through millipore filtration and reverse osmosis, the only bacteria that come through are the water bacteria that we find in our tap water anyway. So this demonstrated by our very elegant technique that the water is indeed safe. And similarly for viruses, occasionally a human virus in the input water, but by the time it has gone through treatment, through microfiltration and reverse osmosis, the only viruses are bacterial viruses not infecting humans, and occasionally a pepper mottle virus, which only affects pepper plants and not humans. And so clearly the viruses are removed with each step of the treatment. There was a concern about antibiotic resistance being transferred to the water bacteria and then increasing in the water supply. But because we can analyze right down to genes, we were able to show that the antibiotic resistance genes are also screened out along as part of the microorganisms in which they are being carried. So there is not a threat then of antibiotic resistance being carried into the natural water supply. We've been doing analysis of drinking water, and we've analyzed bottled water, artesian well water, tap water, and public fountains at various places in the U.S. We were able to determine, characterize different drinking water. For example, in the green dots encircled non-mineral water, bottled water, and the blue is the natural mineral water, and off to the left are the tap waters and drinking fountain water. Now we've been able to show very characteristic the microbiome, so to speak, of drinking water. And that is that it's quite characteristic. You can determine which kind of water by the pattern of microorganisms that can be identified and demonstrated in the drinking water. And so in conclusion, with respect to the water studies, we've been able to show that the predominant bacterial species are very harmless and perhaps beneficial microorganisms in providing taste and structure, a protective structure. The DNA of opportunistic plant and animal pathogens were detected, but not the human pathogens or dangerous bacteria. Let me now turn to COVID-19, and I'm going to pull all of this that I have given to you together. COVID-19 is a pretty, as we now know, nasty infection. It can infect not just the lungs and create problems in breathing and respiration, but it can affect the liver, kidneys, the intestines, and it even can affect the brain. In fact, some severe patients have hallucinations, and we know that there's a conjunctivitis that goes with the COVID-19. And we are aware that that it affects the sense of taste and smell. Now, I'm going to pull all the data together from the molecular genetic, from the environmental, and to develop what we now can determine, and that is we can detect the SARS-CoV-2 virus. We can detect it in clinical specimens, and more recently, studies we are doing to determine the early warning by detecting the SARS-CoV-2 coronavirus in wastewater and in sewage in various communities as an early warning system. Now, in respiratory samples, the SARS-CoV-2 can be detected very easily. In fact, we're able, with our very sensitive technique, we're able to determine even the strains of the SARS-CoV-2 virus. This turns out to be extremely helpful because the virus does seem to change in different communities and also even in individual infection. Now, it's been detected in about 50% of stool samples in the early publications, and there are now many, many investigators looking at the SARS virus, the COVID virus in wastewater samples. The reason is, this looks very complicated, but it isn't. The red is the throat swab positive. If you go down vertically, those are different individuals, and horizontally, it shows the presence. The red, the virus is detected in the throat swab, and then the yellowish line indicates the persistence of the virus in the fecal sample from the given individual. So you can see that for quite a long time in many individuals, the virus is persisting and is shed and therefore can be detected. So SARS-CoV-2 virus has been detected in stool samples of individuals infected with the virus. Studies have been done in the United States, but very early in the Netherlands, and publications from the Netherlands, Sweden, and Australia. And many other papers are now coming out. So the method can be used to measure the effectiveness of interventions like social distancing, because if there's a trend decline in the wastewaters in a community, then the interventions are working. And similarly, if a community doesn't have any cases, but we're monitoring the wastewater, we can pick up the virus perhaps 7, 10, 14 days before the victim actually begins to shed the virus as a result of active infection. So this becomes a powerful tool. Now, there are environmental factors, just as with cholera. In the case of SARS, it was shown that humidity plays a role. With COVID, we now know that that humidity really is another factor amongst many environmental factors that we have been collecting, along with the studies that we had done with cholera in using satellites to monitor movement and migration of populations, to monitor the quantity of water, to monitor the dew point, which can be measured as ground truth. Putting all of this together, we now have reached a point where we can predict, and in this case, this was the first study that we carried out between April 19 and April 30th, the end of April, in Michigan, in the United States. And you can see where our prediction on the left and the actual cases on the right. So this then has become a powerful model that we're in the process of writing up for publication to use as a means of predicting, let's say, upcoming outbreak or resurgence in the fall. A real-time prediction for COVID-19 was in May. That was our most recent. We've now been working in June and July, and will continue to do the analyses and make the data available. So let me conclude by saying that we have had epidemics from the pre-1000-year era, plague, bubonic plague, cholera outbreaks periodically in 1817, 1837, 1881, etc. And now we are going to, I think with coronavirus, very likely have a persistence. Obviously, flu is in the coronaviridae. So we will then, I think, can anticipate that quite likely there will be COVID-20, COVID-21. So with this predictive model, being able to provide early warning in the case of cholera 4 to 8 weeks early, and perhaps we will be able to do the same now with the COVID-19, that we will have the ability to use environmental parameters, molecular genetics, tied together and with massive amounts of data to be able to predict accurately and precisely. So I'd like to close by thanking the many, many investigators in Bangladesh at the International Center for Diarrheal Diseases Research, in Calcutta, India, at the University of Maryland, collaborators from around the world with whom we've been able to build the database. Particularly, I would like to sound out Carla Pruzzo and her team in the University of Genoa, Italy, and our colleagues in Italy, where we were able to do the tracking of Vibrios and the correlation with climate change. 4 people particularly deserve a shout out. Dr. Haq, with whom I have worked for about 40 years, Dr. Antar Jutla, who is responsible for the latest and the most effective models, predictive models using satellite imagery, Dr. Sion Young, who helped build the database that has been so effective in our studies with DNA analysis, and Dr. Hassan, who brings together the DNA analyses and the bioinformatics, along with the team at Cosmos ID, a very effective and powerful team. Water, safe water, is a global challenge. It's not going away. It's going to become even perhaps the challenge for the 21st century as we move into the 2030s and 2040s. I'd like to use the words of John Muir, the founder of the Sierra Club. His words, I think, are really very relevant for us in the 21st century. When one tugs at a single thing in nature, he, and I inserted and she, find it hitched to the rest of the universe. So I thank you for the opportunity to speak with you, and I'd be very happy to take questions.
Thank you very much for this fascinating talk. We have a number of questions from the audience that I would like to read out to you. So the first question is by Dr. Xiao from Harvard, and he wishes to thank you also for the presentation on this fascinating, timely topic. And he's asking whether you have an estimation on the sensitivity of the NGS-based COVID water monitoring.
We're working now in the wastewater between 10 and 100 most recently, and we're working to perfect that. With respect to the COVID prediction, we are, I think, pretty good for the 4-week prediction, and we're trying to work on an 8-week prediction at the moment.
Mm-hmm. And do you have any estimate on what your sensitivity is, as in how many patients or how many individuals would have to be infected in a block or so that you see a signal?
I would love to say we've got it right down to the last person, but we're working very hard on that very, very point, as is about 50 other people working in laboratories around the US and probably many more around the world.
Right. There was a related question by Dr. Wabai from Krakow regarding how you standardize or normalize your quantitative measurements in the wastewater. I mean, if you have more or less rainfall, surely this will give more or less dilution of the samples?
Right now, we're working closely with the sewage treatment plant operators, and we're measuring with respect to flow through the plant, and also with the volume size that we use of the sample collection. And we're also, for the moment, we're working from time of maximum flow and samples twice a week. The game is really beginning, and we're all really scrambling to bring this together. And we're very happily sharing our data with all who are interested.
Oh, that's excellent. There was one more question from Dr. Xiao asking whether you have done similar analyses on any samples that might have been taken but not analyzed, kind of archived, like people have done in Spain from last year before the crisis erupted.
We don't have analyses done yet, and we're working with we're working in the state of Maryland contractually with the Department of Health and Department of Environment, and we just launched the studies there 2 weeks ago, and so we will be asking for archive samples, and we will definitely be analyzing them, because this is a really very, very interesting point, and I think the results from Spain are intriguing, terribly intriguing.
Oh, that's excellent. Thank you so much. There's one more question, if I may, by Dele Ogun Remy, who's curious whether you could say a little bit more about the mutation rate of the virus within patients, because you mentioned that the virus mutates not just across patients, but also within a patient.
Yes, this is a study that is just underway, and it's actually with a school of dentistry. And the question is whether there is an alteration from early stage to end stage. And the data are very preliminary at the present time. But I think this is something that a lot of people are looking at. And I think it's really critical because there is already reports of mutants with not necessarily enhanced infection, but certainly persistence. And so I think we need to pursue this as avidly as we can.
Thank you. I have a question, if I may. I was very impressed by the breadth of this study, and as you closed your talk, you gave this quote: "You tug on one thing, and it's attached to something else." I was wondering, when you go into such a complex question, is what you're looking at driven by mechanistic hypotheses or models, or do you do wide screens as to what type of parameters in climate, in ecology, in these physiological systems, what you're actually looking at? Because potentially you could be looking at thousands of things, right? So how do you start? Do you screen or do you go in with the hypothesis?
Well, first of all, the hypothesis that was initial was that the cholera Vibrio, in the case of cholera, is actually has a host, and it's a vector-borne disease, in my opinion, because the vector doesn't fly; it swims, the copepod. And so from there, we followed the vector-borne models that had been presented. But I must also say that we pull in databases. For example, we work with the British Meteorological Organization. We work with the British Aid Agency, with NASA in the US. And then we have our database for the molecular genetics and genomics. And so there's a multiple series of databases that we draw on, rainfall databases as well. So we are essentially, I guess, dragging in from every direction that we can information, because there's no one parameter. For example, rainfall and temperature were used 100 years ago by the British colonials in India for a very, very, very crude relationship for onset of cholera. Obviously, rainfall remains important, but with all of these other subtle and not so subtle parameters that can be included from databases, we're able to get a far more precise prediction, not only of timing, but also of intensity. In fact, we were doing analyses around Yemen, and we picked up a warning that there would be cholera in Sudan. Now, it wasn't predicted, but we let our British colleagues know. They were alerted, and sure enough, cholera, where we had predicted, showed up. So the model works really very effectively, and we're very excited about it. We've been asked to expand the work to Africa. We had already done some preliminary work with some of our earlier, less sophisticated models in Mozambique and Senegal. So we'll continue that with the improved models and the elegant models we have now. And then we'll also move into some other areas of Africa for predicting cholera.
Thank you very much.